First, I'd like to thank the organizing committee for inviting me here today to talk about dynamic brain imaging. I'm a neuroscientist with over 20 years of experience in Neuroligan PET, and today I'm going to talk to you about what we can do now with dynamic PET imaging, and also what the innovations in PET MR could mean for us in the very near future. And then I'm going to end with a wish list for algorithms and targets. So the definition of dynamic brain imaging is that we're acquiring information about changes in signal in the brain over time. And this isn't unique to PET. There are MR modalities that also depend on dynamic data. However, with PET, what we often are doing is we're observing the behavior of a tracer as it associates and dissociates from its target. And I've illustrated this here with two different dopamine D2, D3 ligands. On the top, we have F18 folipride, shown through here, shown with axial slices from 1 minute post-injection all the way out to almost 3 hours. And on the bottom, we have carbon-11 raclopride, shown from 1 minute to 50 minutes. And although both of these tracers bind to the same target, they have very different kinetic properties. But we can take these information about these kinetic properties and use it to get what we're really trying to understand, which is receptor density. In order to obtain quantitative estimates of receptor density, which is actually more accurately called receptor availability, we extract radioactivity in the tissue, concentration of radioactivity in the tissue over time, as is illustrated in the graphic on the left. We have raclopride time activity curves from an area of the striatum, which is rich in dopamine D2 receptors, and that's in blue. We also have a curve from the cerebellum, which does not have very many D2, D3 receptors, and that's in red. And these time activity curves carry information about the tracer kinetics. And so what we do to get estimates of receptor availability is we feed these curves into a compartmental model, which is illustrated on the right-hand side. And this is a two-tissue compartment model, and it uses differential equations to describe the behavior of tracer over time. So to get out parameter estimates that are related to receptor availability, we solve for the differential equations, and we end up with metrics such as binding potential and volume of distribution, which are two metrics you'll see most often in the literature. And part of this particular conceptualization of our compartmental model, if you'll notice, there is endogenous neurotransmitter bound to the receptors that affects the signal that we measure. And this is going to be important in a few slides. So when these technologies started coming out in the 90s, we all got very excited because it was the first opportunity we had to be able to study neurochemistry in humans in vivo, and the possibilities at that time for neuropsychiatric disorders and neurodegenerative disorders really just seemed endless. The initial studies really focused on receptor availability or trying to quantify receptor density, but that's not the only thing that we can do with these methodologies. We can use PET and reversibly bound ligands to actually image neurotransmission. We can get estimates of whether a neurotransmitter level went up or went down with some sort of challenge. And these challenges could be pharmacological manipulations, they could be cognitive or behavioral tasks or motor paradigms. And this has actually been done quite a bit. If you search PubMed for positron emission tomography and dopamine and raclopride, which is the most commonly used radiotracer for this, you're going to get over 900 results, which seems like a lot of data. But actually, you have to keep in mind that not very many sites can do this work, because you have to have a cyclotron and you have to have a radiochemistry facility. So actually, not that many institutions can do this. What I'm showing you here at the bottom is a typical way of data presentation that you'll find in the literature, which is showing voxels where dopamine was statistically significantly increased in response to a challenge task. And this particular task was presentation of beer flavor. Now, this isn't the only way we can look at data. My colleagues at Yale recently developed a novel approach called Neuroligan PET, or NT-PET, which actually can recover the neurotransmitter release function. And what I didn't mention in the previous slide is that's kind of almost a binary way of looking at it, whether dopamine levels went up or went down, but it doesn't have any temporal information about the dopamine signal itself. So what NT-PET allows us to do is recover temporal information and information about the shape of the neurotransmitter release function. And this is just illustrated with simulated data here. On the left, we have simulated neurotransmitter release curves, which then will result in deflection of the time activity curves as illustrated on the right. And so what NT-PET does, it takes information about the deflection of these curves and then uses it to back extrapolate, if you will, the original neurotransmitter function. And Evan Morris and Kelly Cosgrove have already done some initial work with this in human data. And what they've been able to show is that there are sex differences in both the temporal and spatial functions of dopamine release specifically in response to smoking cigarettes. But regardless of which technique you use to study neurotransmission, there is one huge component that really is problematic for all of us, which is noise. The noisier the data, the poorer the fits are, and the more unreliable the parameter estimates become. So this is a big problem for us. And I've tried to illustrate this a little bit by showing you a time activity curve on the left from these are raclopride time activity curves from a ventral striatal region of interest. So the curve on the left is an average of many, many, many voxels and is pretty smooth, and any fits through that would be probably pretty good. However, on the right, you have a time activity curve from a single voxel. As you can see, it's much noisier. And honestly, that's not even the noisiest voxel level data I've seen. So the fits for that are probably going to be pretty poor, and hence any parameter estimates off of that are going to be unreliable. So because of noise, we spend a lot of time worrying about false negatives or not finding actual effects. We also worry about false positives as well. But typically with noise, what happens is you have smaller effect sizes, and that means you need a lot more subjects to recover effects. And PET is really pretty expensive, and so this becomes cost prohibitive and often makes it difficult to sustain the level of research that we'd like to. And this is why I'm very excited to learn about the upcoming innovations in PET MR technology that hopefully will be available to us very soon. I really think this is going to be a game changer on several levels for those of us doing Neuroligan PET research. Because increased sensitivity is going to mean better resolution and better signal-to-noise ratio. And this is just going to have positive effects through our entire acquisition and data processing stream. It goes without saying, we're going to have dramatically improved quality all the way down to the voxel-wise level. We're going to have better statistical power to detect effects. We're going to have greater stability in parameter estimates. So there's really no bad thing about this here. We're also going to be able to more accurately assess very thin cortical regions and very small subcortical nuclei, which typically we don't have a lot of faith in some of the data that we get with that. So this is really important for both neuropsychiatric disorders and neurodegenerative disorders because some of these small structures are really critical in the pathology, but we haven't always been able to access what the pathology is. And with the right tracers, we're going to have improved sensitivity to detect disease processes as they emerge and not wait till it's too late and the pathology has already done the damage. And this is going to be really important for neurodegenerative diseases in particular. Personally, I would like to be able to use this technology to get data-driven reference region tissues that were not previously identifiable because of tissue heterogeneity and spill-out effects. And if we can have better access to reference region tissue, then that's going to allow opening up the stable of tracers that we already have for example, GABA and glutamate neurotransmitter systems. And it's also going to expand the number of sites that actually can do these, that can use these tracers. I also think that greater sensitivity is going to really allow us to take some theoretical questions into the experimental realm. So one of the things that we get asked quite a bit, usually from electrophysiologists, is how in the world can you possibly reconcile the temporal and spatial specificity that we get with electrophysiology, that we get with fast scan cyclic voltammetry? How do you even try and translate that up to what you guys measure with PET on an ensemble level? Well, I think with the sensitivity of these newer generation scanners, coupled with the cutting edge technology we're seeing evolve with 3D printed organoids, I think we can really take this into a translational experimental realm and start answering these questions. How do we get from micro to mesoscale? I think another advantage we have with this greater sensitivity is we're going to be able to do simultaneous MR and PET experiments that can answer some conceptual questions that are still outstanding in our field, specifically with respect to fMRI and the blood oxygenated level dependent signal. Now we can ask questions about how bold signal relates to neuronal activity, including neurotransmitter release. We can ask questions about what are the various contributions of components that underlie the bold signal in the neurovascular response, such as metabolism and blood flow. And another question that we get asked quite a bit, especially with respect to drug challenges, is, hey, you've got this drug on board, this drug might change blood flow, how does that alter what you're measuring, how does that alter your PET kinetic signal? So we can do, for example, perfusion MR or arterial spin labeling at the same time we're doing the dynamic emission acquisition and start getting at those questions. I have a wish list of algorithms that I'd like to see be made more readily available. One big one is online motion correction during acquisition. And this is really huge because motion is a major problem for both clinical work and research acquisition protocols. It's a big problem for our data quality. Similarly, it would be great if we could have MRI-based partial volume correction at the level of the reconstruction so that we could get better spatial resolution and be more confident in accurately resolving the source of the PET signal. Offline reconstruction would be really helpful. We like to optimize our protocols, sometimes post hoc, to make sure that we've got the best data quality possible. And it also will help us with some data repair too. As far as tracers and targets go, what I'd really like to see is neurotransmitter sensors, because that would be a huge step forward in being able to quantify both synaptic and extracellular neurotransmitter concentrations and also better characterize the dopamine release functions. Some of this work is already being done with MRI, with protein and nanoparticle sensors for dopamine and serotonin, but I think it would be really smart if we could leverage the sensitivity of PET for this kind of technology development. It would be really helpful to have more F18 ligands that are sensitive to neurotransmitter displacement. And this is primarily because I'd like to see the number of research groups involved in this work be expanded. Really just more F18 ligands in general. And there's a big push for this with the clinically approved, FDA-approved F18 ligands for tau and amyloid. But I think it would be great if we could expand this into other realms of neuropsychiatric research as well. Similarly, I would like to see soluble sensors for soluble beta amyloid, because the presence of soluble beta amyloid is thought to be the tipping point for plaque formation and hence one of the hallmark neuropathologies of Alzheimer's disease. If we could detect A-beta-42 peptide versus A-beta-40, that would also be really diagnostically relevant for AD as well. Similarly, tracers that are selective for phosphorylation states of tau would be very useful. There's a lot of dementias that are tauopathies besides Alzheimer's disease, and I'm sure they would appreciate being able to have that accessibility. And then everybody's favorite, targets for neuroimmune responses. The tracers that we have available to us for this now are suboptimal, but getting really good tracers is a very complex problem because there's multiple phenotypes of microglia, which are the primary targets of interest for this. And on top of the multiple phenotypes in microglia themselves, these phenotypes actually are divergent across different disease states. So you have an additional layer of complexity on top of that. And unfortunately, in the field of glial biology, the pharmacology really hasn't gotten there yet for us to use that as a springboard for radio labeling. There's a lot of targets there that have been identified that are very, very much of interest, but we haven't TREM2 is a really good example of that, but we haven't identified the endogenous ligand, and there's no good pharmacology yet available. And with that, I will end, and thank you for your attention and for your time.